Today, what I'd like to do is we're going to have an open forum. So we try to keep this very informative, but yet very casual. We will not be presenting any data slides or anything like that, so the focus will be on the panelists because these are the experts. And today what we'll try to answer questions about are some new investigational therapies in pulmonary hypertension and stem cell therapies for pulmonary hypertension. And sitting with me today is Dr. James Ford from the University of North Carolina, Chapel Hill, Duncan Stewart from the Ottawa Hospital in Ontario, and Scott Visovatti from the University of Michigan in Ann Arbor. So I'll start. My name is Ray Benza. I'm from the West Penn Allegheny Health System in Pittsburgh, Pennsylvania. I've been treating pulmonary hypertension for 20 years now. My interest in the research lab is in the area of genetics and pharmacogenomics. Learning, and basically what pharmacogenomics is looking at an individual's genetic composition, looking at the variations in the gene sequences, and then determining if those individual fingerprints can determine whether someone will or will not respond to a particular medication. We have a very large National Institutes of Health initiative that has funded this work. 1,500 of you have participated in this trial, and we have a lot of information, and we likely will be bringing a lot of this information to the public arena in the next year or two. So a lot of information to do. So we'll just kind of go down the panel, introduce yourselves, and then tell a little bit about the work that you do.
All right, sure. As Ray said, my name is Jimmy Ford. I work at the University of North Carolina in Chapel Hill. I've been involved in pulmonary hypertension for about 7 years now. Particular clinical areas of interest with regards to pulmonary hypertension at our center are patients who have sarcoidosis-associated pulmonary hypertension. We've been involved in 2 open-label trials looking at the efficacy of certain therapies for that patient population. We also have a very active liver transplant program, and I see a lot of portopulmonary hypertension as well. We've been doing some biomarker studies at our institution, also looking at the differences in the inflammatory and procoagulant biomarkers in the pulmonary circulation versus the systemic circulation. And we've also been involved in a few industry-initiated trials looking at some of the investigational therapies, which we'll probably talk about today, particularly nilotinib and cyclotinine.
So I'm Duncan Stewart. I have been involved in, I'm a cardiologist, been involved in pulmonary hypertension and pulmonary hypertension research for a number of years. I don't like to say how long, probably 20. And it really began when we were playing with endothelin-1 in the early days after its discovery and recognized that it was a key player in pulmonary hypertension, which obviously led to some of the therapies that we have now. And since then, I've been very interested in trying to find better ways to deal with this very difficult disease. And over a decade ago, we began to explore the possibility of using progenitor or stem-like cells to try to enhance repair of lung blood vessels in the lungs. So that's been a big part of what we've been doing since then. And as many of you know, we started at an early phase clinical trial, which we're going to close out soon, looking really at the safety, and we hope to move forward into further clinical testing of various stem or progenitor cell therapies for pulmonary hypertension.
Hello, my name is Scott Visovatti, and I'm a cardiologist as well at the University of Michigan. I'm primarily a basic science researcher, but have a real clinical interest in exercise physiology, and primarily using that in the heart catheterization lab. My research interest really has to do with small particles that circulate in everybody's bloodstream, with the possibility that they carry with them or inside of them proteins, or what I'm mostly interested in is enzymes, which could perhaps contribute to the development of various forms of pulmonary hypertension.
So interestingly, as many of you heard yesterday in the conferences, the evolution in therapy for pulmonary hypertension has really shifted paradigms from using medications that we thought would just dilate the blood vessels to now medications that actually restructure the blood vessels to allow them to be a larger caliper to promote better blood flow. And in even more novel therapies, such as what Duncan is talking about, is to actually try to replace some of those cells with cells that are normal or don't have specific genetic defects, and to try to allow those cells to take over the operations of the blood vessel and basically to teach the blood vessel how to be normal again. And then all the way to the most advanced therapies right now that we're looking at, which is regenerative therapy, as I heard Greg Elliot talk about in that beautiful talk that he gave last night, in which we are taking lungs, we are decellularizing them, leaving just a skeleton of the lung, and then reinfusing them with normal progenitor cells, the basic cells of life, and these cells will reconstitute these skeletons and basically produce another lung that we could then use as a transplant model for giving back these normal lungs. That is kind of from reality, which is these remodeling medications, to stem cell therapy, to really the pie in the sky, which is this regenerative medicine. So really with that framework, I'd like to open up to the audience questions. We try not to deal with very specific patient-detailed issues, so if there's a problem with your medical therapy or if you have something about your own medical therapy, we try not to deal too much with that, but general questions about the therapies that you're on, the new therapies that are coming up, how they work, particularly the interest in these microparticles and the stem cell work and the regenerative work. We'd be happy to answer any questions dealing with any of those topics. Sir.
Hello, my name is Alex Salama. I'm interested in finding out where is Dr. Duncan's research stands at right now, because I attended one of the sessions, I think, in California, and you had a beautiful presentation, and I wanted to see how far you progressed so far.
Great question. Thank you.
So thank you for that question. It's been a slow journey, and it's not surprising. I guess it's not easy to do. So I think we're basically completing this small trial that we've done, which has really been a single site in Canada. We've not been able to enroll patients outside of Canada for various reasons, and we're now working very hard to move it into a bigger effort that will involve, in fact, mainly be in the U.S. and will involve a number of different sites. So we're working with a industry partner to do this, because you need that kind of help and resource to get these things going. And we're really preparing now to submit that to the regulatory folks. And we're looking at each step along the way in terms of the cells we use and how they're processed and how we manipulate them. Because in our therapies, you know, we have included a genetic engineering approach, and that was based on our earlier preclinical work in the animal models, that we felt you needed to really do something to increase the efficacy, the ability of these cells to do what you want them to do, particularly in the case of established disease, when if you're trying to take an animal or a human, for that matter, that has severe pulmonary hypertension and then turn back the clock and reverse it. This is not a trivial thing. And based on our experience, we thought, well, cells, they do something, but they need help. So we work on, there's probably lots of ways you can do this, but we have done a lot of work on this genetic engineering approach. And I think we're going to persist in that. But that creates, as you might imagine, a few complications and questions that the regulatory people are going to be interested in. So we have to do a lot of our homework. We have to get all the data that's going to be required from safety and efficacy and so forth. And we're in the process of doing that now. I can't put a timeline on it. I mean, it wouldn't be very meaningful. I mean, it's not going to be, we won't have that done by this year. I'm pretty sure of that. We're hoping to get it done by next year. So what we're working on.
So I have a question, Duncan, that the cells that you're infusing, how do they know where to go?
Well, this is the beauty, actually. That's when we got started. I'll let you into a little secret. I mean, this has evolved, right? But to begin with, the idea was really to do a gene therapy. And the question was, how do you get the genes where they need to be. And where we wanted to get the genes was in the very smallest blood vessels in the lung. And you know, with viruses and other things, it's very hard to get a very selective target. And so it's actually, I was at one of the early days with Flolan when they were training groups to how you deliver it. And I was thinking, well, you know, this is very complicated. You put in a catheter, you infuse, you have to do this continuously. It's a very cumbersome. Wouldn't it be neat just to have cells that you could stick into the lung and they would be in the right place, just producing it just where you need it, right? And that's when you thought, well, maybe you could just take a cell like a smooth muscle cell or a fibroblast and stick it into the circulation, and it would end up there because that's where the cells are filtered. The lung is a filter in the body, right? Anything bigger than a red blood corpuscle gets filtered out in the lung, and it gets filtered out at the precapillary artery, the arteriole, where we think the disease for pulmonary hypertension begins. So you use the cell really as a vector because of its size, that it gets stuck there. Question is, you know, you got to get it to stay there, and so that's the trick. And fibroblasts actually are very good at staying there. So we did a lot of work with them initially, and they stay there quite nicely. But of course, we then moved on when we began to understand that there were cells that actually had regenerative capacity in their own right. It became very logical to try to use both a regenerative cell and then try to enhance the gene therapy. So that was the evolution. So it really is the cell that gets us to the right place because that's where it sticks.
So you're creating local bioreactors in the lung to create chemicals and substances that teach the lung how to behave normally again.
Yeah.
Right, very good. Ma'am, you have a question?
Kimberly Ford, what are the steps or stages of a study?
Jim, wants to handle that?
Yeah.
Sure. That's a loaded question.
Be careful.
Are you referring to clinical trials in general? So, I mean, generally in the development of a medication, it's initially looked at in a laboratory setting, animal studies, looking at safety and efficacy in either mouse or dog or sheep or other animal models of pulmonary hypertension. And once some sort of signal is identified with respect to effectiveness, then small initial studies are done in humans looking at safety and efficacy first, principally. If once signals are seen in those smaller groups, we progress to larger clinical trials. So that's kind of a general overview, proceeding from animal model with a first thought towards safety, and at least some signal of efficacy, and then moving towards larger trials.
So there are basically 5 phases of development of any therapeutic that we use in the human body. The first, as Jim was alluding to, are called preclinical trials. And those are trials basically in animals to see if the mechanism is right, if the drug kind of does what it's supposed to do, and most importantly, it is not toxic to the animal. Once that is established, the drug then goes to something called a phase 1 trial, and that is conducted in humans. And a phase 1 trial is basically trying to get the right dose of the medication to use for subsequent trials. Once the right dose is assessed, and basically these very early trials are always looking at safety, a phase 2 trial is conducted. And the main purpose of the phase 2 trial is for safety, to make sure that the drug does not hurt people. Once that's established, then we progress to the phase 3 trials, and those are the trials that I think many of you have participated in which we see if the drug works, if it does what it was supposed to do, if it worked just as good in humans as it does in animals, and that's where the approval process of a drug usually will take. Once you've passed the merits of the phase 3 trial, then the company will approach the FDA, and the FDA will review all the data to make sure that they think that the trial was conducted in an ethical and meaningful manner, and then approvals will be based on that. Now, your particular participation in a trial is even more important to that, and I want to remember everyone in the audience to remember that participation in clinical trials is a totally voluntary state of being. There should be no coercion in participating in a clinical trial. These trials are meant to make progress, but it's all up to you. You guys are always in the driver's seat. Always remember that. You are always in the driver's seat. Never feel like you're pushed to participate in a clinical trial. Obviously, participating in clinical trials we think is a great thing because that's the only way these new therapeutics will come to market and be useful in treating this disease. So we encourage participation, but you should remember that participation is always voluntary, and the informed consent that many of you have signed for trials is a critical process in ensuring that voluntary nature to make sure that you understand exactly what you're participating in. So that's a very, very important document, not to be treated lightly, and you should really take time, 20 to 30 to 40 to 50 minutes, even overnight, to read those consents to make sure that you have a very good understanding of what the clinical trial's purposes are.
I have a question. I'd like to go back a little bit. I'm right here. Sorry. When we were talking about the cells going right to the heart or the lung where it needs to go to reproduce and to correct the cells, I've read some information and I'm wanting to clarify. Are we at the point where the cells don't know when to shut off?
Well, yeah, it's a very good question. I mean, and this is all the speaks to some of the safety issues, especially when you're genetically engineering the cell. So, I mean, it's a very good question, and it's one that we've obviously thought about. So, first of all, the way that we engineer the cells is temporary. So we're not using any. So we use just very simple plasmid DNA circles that so with no virus involved that get into the cell, and these don't actually incorporate into the genetic material of the cell itself. So they hang around outside of the host genome, and they only stay there for days or weeks, so maximum 1 week or maybe a little bit more. And then that genetic material the cell actually has mechanisms of getting rid of that, and it disappears. So the gene enhancement step is a transient step that will only help the cell do what it needs to do within the first week after it's delivered. But then again, the cell, it's the patient's own cell, so we're doing what's called autologous cell therapy. So we take the blood cells from the patients, and then we isolate and manipulate them and then give them back. So it's the patient's own cells. So that should be reasonably safe, but you never know. And interestingly, again, even those don't stay in the lung as long as we thought they would. So in the first week or so, they do stay maybe 20%, but over time, they leave the lung. So we had thought they would permanently persist within the lung, and that doesn't seem to happen yet. At least in our preclinical testing, we get very strong effects even with that transient residence of the cell. So that's opened up a whole bunch of questions as to how these cells actually do what they're doing. And we've moved from the idea that these cells actually produce new blood vessels themselves to the idea that the cells probably release factors that induce the repair of blood vessels within the lung. Or they release other things like the microparticles, and that these may be involved in some of these repair steps. So all that is interesting research. But from a safety point of view, actually, it's a good thing, because if the cells and the gene aren't staying there for prolonged periods of time, there's less chance for that to cause problems.
Yes.
I'd like to ask, like everybody would like to know, we all appreciate your research and what's happening, and this is a great effort. But how soon do you see the cell development and actually affecting or helping people in this room in their lifetime? Did you see this? Is there any promising stage 3 or studies that are going on right now that look like they'll be useful to people while we can still, take a benefit from it? And the other question I have is, a lot of people would volunteer for trials, but I volunteer for some myself, but they always say, well, you're too sick. You've got too many morbidities, problems. So is it hard to get people to volunteer to be test patients or guinea pigs?
Yeah.
So we'll answer the second question first, and then I'll let Duncan handle that first question. It is difficult to enroll people in clinical trials because within the framework of a trial are certain inclusion and exclusion criterion. And unfortunately, these criterion are often very strict and because we, as a community, feel that the best way to get a good drug with promise to people is to test it in a group of people that have very, very similar profiles and that strictly follow the definitions of the disease that we're treating. Because often those are the people where true response can be noted, particularly a favorable response. And once those favorable responses are noted, then we can and when the drug is approved, then each individual physician has to make a very conscientious decision on whether that drug, which has been approved and tested in a certain group of patients with very like features, can then be extended to patients who have comorbidities, like you mentioned. Because the comorbidities bring in a whole other nuance to the science of using that medication. We have to make sure that the new medication doesn't interact with any established medications, that the new medication doesn't actually interact with any of the comorbidities. So it is a very conscious decision that has to be made at that point. And unfortunately, people with a lot of comorbidities are often not included in clinical trials. But then in latter work, and sometimes we call these phase 4 trials, in which you bring a drug approved for certain subsets and tested and extended in people with comorbidities to see if it works in there. So in these phase 4 trials, people with some of these comorbidities are included and seen if the drug is efficacious. Now, as far as the stem cell therapies and are they actually in the clinical environment right now, I'll let Duncan attest to that, but the answer is yes.
Yeah, but in a very small way. In pulmonary hypertension, there's much more activity going on for other diseases, particularly heart disease, myocardial infarction being the indication where there's been the most testing. Nearly 2,000 patients have actually been enrolled in similar cell therapy trials for that condition. And so we're beginning, and a phase 3 trial actually is starting this fall. So we'll have some information about that as it relates to a different indication. And we're watching that very closely. In terms of pulmonary hypertension, it's been slower. It's not as widespread a problem as heart attacks. I guess it's been, part of the problem is it's a very new field, so it's taken a while, I think, for the industry to recognize this might be something that they can develop as a viable therapeutic. So it's been I've been working for 10 years on this, and it's really been a struggle to get the resources to do this in the way that you have to. And as you mentioned from your first question, because you're limited, especially when you're starting with such a novel therapy, you really have to limit your enrollment to very specific kinds of patients, and that will be comparable, that you can really understand understand the safety, for example, of the treatment, and that means that most of the people that would like to have therapy aren't actually eligible. So enrollment has been a huge issue for us. So we've learned a lot about that. So that's slowed us down a lot, just because accessing the number of people that we need, and given that we can only do it in 1 or 2 sites, given we're restricted geographically, and given that we have very restricted protocols. So, but I think we've learned a lot doing that, and as I say now, I think the field's moving to the point where we can start doing it a little bit, the way it needs to be done, more broadly, with more sites being involved and more resources brought to bear. And then that lays the groundwork for doing the next phases of testing that you really do to be able to offer it more widely. So I'm thinking, if we're successful in moving this forward, it could be in 2 years we'd be doing a phase 2 type trial, really looking at whether it works, and then it all depends. If it works well, then I think yes, I could see this moving forward to being available more widely within 5 years, for example. Yeah, absolutely, and I'm glad you brought that up. The problem, of course, is that that's outside of any of the kind of regulation we're talking about, and that regulation is there to protect all of us. If in this kind of stem cell tourism, nobody is checking what is being delivered, nobody's checking whether or not it works, no one is checking whether or not it's safe. The concern is that in fact, I think it's pretty likely the main motivation of many of these organizations is commercial. They want to take your money. And so, they're not publishing their data. They're not providing it so that anyone can look at it. We're learning nothing from this. So we don't know whether the patients that spend $50,000 to get the cells are getting good product, whether they're doing better or not, whether there's safety issues. So, that kind of thing is not going to help the field.
Hi, I have a question about where you get the stem cells from. I've heard that it's from abortions.
So I can't see where you are.
This is right here.
Oh, okay. Always disconcerting trying to answer a question when you can't see the question. That's right.
I've heard they've come from either abortions or maybe the blood from the umbilical cord.
Yeah.
So I'm just wondering, where do you get the stem cells from?
Okay. I'm glad you asked that question. And you can get them from, for various purposes from all different sources. In our case, we're taking it from the patient themselves. It's just from the blood. So this is autologous cell therapy. So we do more than just take a sample of blood, though. We do what's called an apheresis procedure. So it's a machine not too dissimilar to a dialysis machine, I guess. You get attached up to it for a few hours, and it basically skims off the white blood cells out of the blood. So and returns all the rest of the blood back to the patient. It's your own cells, your own blood. So now whether that's the best way to go in the future, it's a question, but there are advantages to doing it that way. Your body recognizes them as your own. From an ethical point of view, there's not a big question about that. The problem with doing your own blood cells is they have all the same problems that other cells in your body may have. So, for instance, if pulmonary hypertension is caused by a gene mutation, for example, then the cells will also have that. So that's an issue for this kind of therapy.
And remember, everyone in this audience has stem cells. We all have progenitor cells that circulate within our blood, that reside within our own tissue. It's just the type and the quantity that differs from person to person, that changes as someone moves in age from younger to older. And in someone who has comorbidities, it changes the character and nature of the type of stem cell into one that may be a good quality stem cell or a poor quality stem cell. So the use of our own stem cells to create and fix conditions within our own bodies, they're a novel concept that we're trying to capitalize on.
Okay, one more follow-up question right here. So you touched on the point where if you use your own blood cells, because I was going to ask, because we were taught or instructed that PAH is potentially a blood disease that is causing that proliferation. But what I'm trying to understand is, with this therapy, are we regrowing the veins inside the lungs, or are you instructing the cells to produce more nitric oxide?
Yeah. So the idea when we began was that we would regrow the vessels, and that would sort of regenerate the vessels. And we do in our animal models see evidence of at least a restoration of the more normal circulation within the lung. But it's becoming clear it's not the cells themselves that become those blood vessels, but they probably release factors which instruct the tissue around it to be able to make that kind of repair. And nitric oxide produced by the gene that we insert, which is the endothelial nitric oxide synthase gene, probably plays a role because we know it's essential for new blood vessel development. So it plays, in addition to being a vasodilator, it also guides blood vessel development, and very important for the effects of genes like the vascular endothelial growth factor, the classical angiogenic or blood vessel growth factor. So we don't know for sure, but we think that repair or maybe even regeneration is important, but it's an indirect effect of these cells. But we think that's really how they're acting.
What I'd like to do is maybe take one step back for a second, because the issue in dealing with stem cells, I think, is very fascinating, but I also want you to recognize some of the other areas of investigation that are currently going on in this field. And as we mentioned earlier, there's been a shift in paradigm from using medicines that we classically think will dilate to the blood vessels to medicines that maybe remodel the blood vessels. So what are we doing in these areas? And I think we can broadly divide these areas into the following. We know that there are generally 3 pathways that we have made use of in the current therapeutics. So using that as a basis, the scientific field has moved in several different areas. 1, can we use drugs in those 3 pathways that are better than the drugs that we have right now? So has there been development in capitalizing on those pathways that are better than what we're doing right now? The second is, are there totally novel pathways that we have not made use of before that we can now make use of that may work in conjunction with or better than the pathways that we've already made use of? And within that framework are the medicines that we're using now that actually restructure and remodel drugs. Because remember, this disease is one in which cells divide and proliferate and constrict when they're not supposed to. So this really is analogous to a type of cancer. We have uncontrolled cellular growth within our bodies. And so can we use drugs that are used in the cancer arena that capitalize on stopping cells from growing and moving in the wrong directions and use that to control this unchecked growth in the lungs? And then yet the third area of investigation right now is, wow, we have a lot of toys that we're playing with right now. Do we really know how to use them all together? That's very important. We have established therapies right now. Do we know how all these drugs work together? There are better ways of using them than what we're using them in. And then we're talking about the broader therapies like stem cell therapies and regenerative medicine. So I'd like to focus maybe a little bit on these first 3 topics, and maybe what I'll do is I'll have each of the panelists pick one of those 3 areas and maybe tell us a little bit about what's going on in those areas, and then when they're finished, maybe we could take some questions on that. So want to pick a topic that you're comfortable with and
Sure.
expand on one of those 3?
Right. You had mentioned approaching PAH almost like cancer. And one of the drugs you were alluding to was a class of drugs called tyrosine kinase inhibitors. Those are currently approved for the treatment of certain forms of leukemia. And basically, each of them have a certain group of different tyrosine kinases, which are chemicals which control cell growth, proliferation, and the death of certain cells that is intentional on part of the body. They also inhibit something called platelet-derived growth factor, which has been shown to be elevated in patients who have PAH. There have been 2 of these drugs that have been studied in pulmonary hypertension. One of them is called imatinib, which goes by the trade name of Gleevec. And studies done with that agent suggest that it may be beneficial to patients, particularly who have very advanced disease or very severe disease, despite being on some of the existing PAH therapies that we have. So there are ongoing trials with imatinib or Gleevec currently, expanding upon the data that was accrued from smaller studies. Another agent that's currently in study is called nilotinib, which is just has a different affinity for different tyrosine kinase receptors. And that is currently the first cohort of that study was just recently finished, and is the second cohort, which is going to be expanded to many more centers, is set to start within the next few months. And that is going to focus on patients who have a very high pulmonary vascular resistance. So the resistance to blood flow is extremely high in this group of patients, because that is the group of patients where it was thought the most benefit was seen with the imatinib, which was the first tyrosine kinase inhibitor studied. So I think that's kind of where we stand with that.
Yeah, so these medications, which are designed to remodel the pulmonary arteries, the first compound, imatinib, has actually finished its large phase 3 trial. The results that were presented at our recent meeting at the American Thoracic Society several months ago. The results look positive for people with very, very severe disease, but also some side effects were noted in the trial. That drug is being evaluated by the FDA, and we should know something about this drug within the next year or so. So this could be a compound that may come to market, that may be used, and you may be approached by your physician, or you may want to inquire to your physician about this in the next year or so. What I'd like to do is maybe take a little bit of time and talk about the three pathways and what new drugs are coming down those pathways, because many of you in the audience may actually be participating in some of these clinical trials. So as you know, there are three main pathways that we utilize right now. One is called the PDE5 pathway, which makes use of a chemical called nitric oxide. It's really the basics of that system. We have the prostacyclin system, and then we have the endothelin system. Now, within the endothelin system, the established medications that we have right now are bosentan and ambrisentan. Well, there's another compound that has just finished its phase 3 trials called mazasentan, and that is a drug that capitalizes on this endothelin system, but has a newer mechanism of action. It actually works on the intracellular level, and the results of that trial are going to be published very soon. And we'll see if that is a promising new drug that can be used in pulmonary hypertension. In the PDE5 pathway, we have drugs that are actually working at more proximal levels in that pathway. One of the new compounds is called riociguat. And maybe some of you have participated in the patent trial that was involved in patients with pulmonary hypertension. Well, the patent trial is now finished enrolling patients, and the results of those trials are being analyzed to determine whether this new drug, which acts more proximally in that system, will be of any benefit or more benefit than the current drugs that are in that pathway right now. In the prostacyclin pathway, obviously, we have done investigational therapies on new ways to deliver prostacyclins. We have pill forms now that are being actively looked at in our investigation. One of these new compounds is an oral form of Remodulin that has finished their phase 3 trials. The results have been published, and this drug is being looked at by the FDA as a potential drug to bring to treatment for pulmonary hypertension. There's a new analog that instead of giving prostacyclin, we are actually giving a drug that will activate the receptor where prostacyclin actually works. It's a compound known as selexipag. And that drug is currently in the phase 3 trials, as we talked about earlier. We don't have any preliminary results, but in the phase 2 trials, it looked fairly promising. And the results of that trial should be done within the next year or so. So we have new compounds in each one of those 3 pathways that have been actively investigated or at the end of their phase 3 trials. We should have the results within the next year or so of these trials. And if they work and if they're good, they'll be hopefully out in the market within the next 2 years. So, Scott, you want to talk to a little bit about how we can play with the medications we have already, or what's going on with that?
So Dr. Benza had alluded to the fact that part of one prong of this clinical trial approach is really taking drugs that we already have and using them together. Common sense might indicate that, well, if one is good, 2 are better. But as he indicated, there can be interactions and maybe too much of a good thing. So there have been some exciting trials going on recently that involve most combinations that you could think of, combining oral therapies, both the endothelin receptor antagonists and the PDE5 inhibitors, with the parenteral the medications that you receive through a pump. Again, many of these look promising, but we don't have a whole lot of final data on them. Some of them are following up on earlier trials, which in fact have shown benefit. So I think this topic of combination therapy is very, very important, and I truly do believe that it is the way to go. When you look outside of pulmonary hypertension, you look at patients who have high blood pressure, for example. Options often include, well, I start a medication, the patient got a little bit better, but we're not quite where we need to be. What do I do now? do I continue to increase the dose of that one medication, knowing that medications tend to give more and more side effects the higher you go in a dose? Or do I add something else, something that works in a slightly different way to give the same effect, but with fewer side effects? And so I think that's where we are now. And I think it is one of the benefits to being seen, to at least having some aspect of your care administered through a pulmonary hypertension center or specialist. Because not having a lot of these trial data available at this point, I do believe that working with somebody like Dr. Benza, who's been around and seen it all,
oh boy.
You can really derive great benefit when somebody like him says, well, you know, my sense is that you might do better if we bring this and that together. Again, with that idea of maybe minimizing side effects and maximizing benefits. So even though we're saying, yeah, we don't have a whole lot of data on these trials, that doesn't mean the FDA still allows us to carefully use combination therapies as long as the person doing so has experience in that area. So I think that's a very exciting aspect of the future, combining the new drugs with the old. To see what we can bring to bear. As the gentleman in the back pointed out, it can just be an agonizing wait for many of the newer therapies. So it is exciting to try to work with what we have right here and now, whether they're therapies that are in our own field or borrowing from oncology.
Just to add to that, one of the other things I think of avid interest to us as investigators is, you know, this is a bad disease. We all know that. And this is a disease that progresses very rapidly in some people. So one of the approaches that we are trying to investigate now is whether we should hit hard in the beginning or whether we should be using stepwise approaches and adding medications if people don't approach goals. So one of the big initiatives that is going right now in the field is a trial called the Ambition trial. And the Ambition trial, the whole purpose of this trial is try to determine, well, should we be starting 2 drugs up front to hit this disease as hard as we can to stop it in its tracks? Or should we be doing it the way that we have been doing it, is start one medication, wait 3 or 4 months, see if you've reached goals, and if not, add a second drug. So these 2 types of approaches are being used in an investigational manner right now to try to give us a little bit more information on how to use the drugs that we have already in a better fashion. You have a question?
So my question is, there are physicians out there that are using combination therapy that are not in the trial setting. So is that information that those patients are on combination collected in the registry, and who runs the registry? So, because how do you know if that data is going to be helpful for the clinical trials that are currently going on with combination therapy?
And that's really a superb question. This is exactly why trials like the Ambition trial have been developed by the scientific community, because there is a lot of use of combinational therapy right now. And again, as Scott mentioned, it's intuitive to try to use drugs with different classes of action together to see if we can get a beneficial response. And if you look at the registry data, and one of the largest registries we have in the United States is the Reveal Registry, we have some information on that right now. We haven't analyzed it fully, so I can't tell you from a registry perspective if it is more efficacious using that type of approach. But we do have information about that in the real world that we can look at and we can analyze. What we do have is we have a number of clinical trials that have been completed with combinational therapy that have shown that drugs can be used together, and when one drug is added to another, you do get a beneficial effect. So there is now precedence for using combinational therapies, particularly in stepwise approaches, that may be beneficial and may add additional improvements in the way you guys exercise, the way the right heart operates. So there is some credence to that now. But I think the more literature we add on that, the safer we can feel and the better we can feel about telling you that this kind of combinational therapy will work.
I think one other good thing to point out is that kind of out of necessity, a lot of our current trials, even looking at new therapies, have kind of become combination therapy trials in the sense that a lot of patients who end up getting referred to pulmonary hypertension centers where these trials are ongoing may already be on at least one PAH therapy, right? So if we eliminate anyone who's already on a PAH drug in the current era, that would eliminate a lot of potential clinical trial participants. So a lot of our current trials looking at new therapies do allow for some what we call background therapy and have given us some added insight in that regard as well.
If I could bring up a point as well, we've talked a lot about medications as part of a trial, but the registry issue is also an important aspect. In other words, your physician may at some point present you with a possibility of participating, not exactly in a trial, meaning there's no medication here, but a tool that allows us to gather information. And I just want to mention the importance of this. Dr. Benza mentioned the Reveal registry. Many of these help us figure out if we're doing a good enough job as physicians. Are people being at least considered for advanced therapy if they get sicker? Are they being evaluated correctly from the start? Are they, one of the big themes, and perhaps the biggest theme of this conference this year, is early diagnosis and not missing a diagnosis early on. How good are we at getting people where they need to be? So just a plug for the importance of again, as Dr. Benza said, there's no pressure, of course, whether it's you divulging information about yourself as part of these registries or participating in a medication-based trial. But we are just so indebted to you when you do participate in these registries, because it's truly how we get better at what we're doing.
Questions?
Could I just make a comment on the some of the newer anti-cancer medications, which is very exciting, but I think one of the things that's limiting here is we're borrowing from drugs developed for different diseases, leukemia and other cancers, and we're just saying, well, maybe they'll work, hopefully they'll work in pulmonary hypertension. And what we really need to do now is to do the same thing that's been done in cancer, which is taking the cells and then looking at how they're wired and what's wrong with that, and how we can develop drugs that will hit the right switches that are overactivated or not turned on properly in these cells. And this is another way in which stem cells can be useful, because it's hard to get cells from lungs, but you can get, for instance, cells from blood quite easily. And if you can, there are several ways you can go. It's a whole new field called inducible pluripotent stem cells. You can take any cell and turn it into a real stem cell, and then you can differentiate it back into the tissue, lung tissue or vascular tissue that you want to look at. But even easier in pulmonary hypertension, we can take our progenitor cells from the blood, and we can differentiate them quite easily into endothelial cells. So they become identical to endothelial cells. And many people think the endothelial cell may be the starting point of this disease, that triggers the rest, and abnormalities in the endothelial cells trigger all the things that happen later. So then we can take the cells from patients with pulmonary hypertension versus normal people, or patients with mutations that we know cause the pulmonary hypertension, and then we can do very detailed mapping of the genes and the proteins that are involved, and even their activity, whether they're turned on and turned off, just like our cancer colleagues have done. And from that, we hope that we'll be able to then get a map of the activity of these cells and figure out which of these pathways, if it's tyrosine kinase pathways we're interested in, which of them are aberrant in these cells, and maybe be able to design the right kind of treatment. So that's looking way in the future, but already it's showing interesting results that may be very relevant for drug discovery.
When you were mentioning some of the new drugs and pathways that are being looked at, the last one you mentioned was the new prostacyclin. I was wondering what the delivery method they were looking at for that would be.
That is an oral drug that is not really a prostacyclin per se, but it is a drug that will act where the receptor for prostacyclin is, so it directly activates the receptor. So, but it will be an oral medication.
Dr. Benson, you had mentioned earlier, you touched base with the ambiguity with regard to how you would approach duplicate medicines and so forth for a patient that maybe wasn't doing as well, but for a patient that's mostly stable, is there any type of shelf life of effectiveness for these medications? In other words, if a person were doing stable on, say, inhaled therapy, is there any benefit to moving on to the infusion-type medications or these newer ones as they come out? Or what's your opinion on that?
That's a great question. And I guess it really depends on what your personal opinion about stability is. Someone could have very advanced symptoms but be stable with advanced symptoms. And then there are others who can be relatively asymptomatic without symptoms and be stable from that sense. Where I think the field has progressed in a significant level is in being able to tell patients who have these symptoms where they are kind of in the spectrum of the disease, and then based on that determine if more advanced therapies are indicated. As many of you know, there have been several scoring systems that have developed over the last 2 years, both here in the United States, in France, actually two developments here in the United States and one in France, in which we take a collection of clinical parameters, how far you're walking in the 6-minute walk test that we drive you guys crazy with, how well you do on the heart catheterizations, what your echocardiograms look like, what are some of the biomarkers that we routinely check in your blood, how do all those paint a picture of where you are in your disease state, and we're using those now to develop scoring systems. So someone with a stable score of 10, for example, in the REVEAL score, would be a lot different than someone with a stable REVEAL score of 3. And that person who has a higher score, even though their symptoms are stable, we know what the course of someone with that score will be over the next 365 days. And based on that knowledge, we would advance that person's therapy if they were on maybe 1 drug or 2 drugs or try something different. Whereas someone who has a very low score, who we know will have over 95% chance of survival, we may not do anything with that. So I think trying to do the relative risk stratification of people will help us decide when to advance therapies in someone who really does have stable symptoms. It just depends on where in the paradigm you are with that stability.
I have a question. Regarding the stem cell research, I am just wondering, as a PH patient community, is there any information that can be gathered from the community that would help you along with your research? And also, regarding resources, such as financial, is there anything as a PH community that can further progress the stem cell research area?
Thank you for that question.
Well, thank you for that offer. I mean, it's obviously it's difficult to do, and certainly the interest of the community is very important. And when we get to the stage where we're able to have trials running, for example, in a number of different centers, obviously it's going to be very important that patients with this disease be willing to join these trials. And again, it'll be a totally voluntary process, and knowing that when you get into this kind of research, the risks probably are higher than for pharmaceutical research, which we know better what the risk benefits are. So that's certainly important for us. And in terms of financial, I mean, I think we've done largely what we've done is what investigator-driven, largely funded by different sources. Some of it foundation sources from donors that have given. So that's been very helpful. But really, to make this to be efficient and get this, if it turns out to be effective enough that it should be developed as a more widely available treatment, we need industry to be involved in this. They're the ones that know how to navigate, and so I think it's been hard to get that traction, but I think now the world is changing, and I think a number of different companies are beginning to recognize that the future of medicine will involve these kinds of therapies.
You know, remember, everything, you're in total control. You have control over everything. Companies work for you. We work for you. And so the ability for you to reach out for the people who are working for you, I think, is one of the most important steps in moving in that direction. You should be able to call industry representatives and say, hey, what are you doing? What are you working on this for? And, is there something way that we could push these through? People are very responsive to those type of things. And then, working from the very bottom, the PH community has always been incredibly generous in contributing to the research that drives this field. The research room is a classic example. We have several groups in there already have enough samples to do all their experiments with, and it's only the first day and a half of the conference. It's really an amazing thing that this community does. There are other major initiatives. One of them is the Pulmonary Hypertension Breakthrough Initiative, which is a large initiative in the United States that's been funded by the American Heart Association and also by the Cardiovascular Medical Educational Research Fund. And this is a wonderful, wonderful program in which patients with pulmonary hypertension who are undergoing lung transplant at the time of their transplant actually donate their lungs to science, so that after they are a transplant recipient, we use their lungs. And these cells that Duncan has been talking about, these transformed cells, are really derived from this tissue that we have obtained. And that we've learned that from doing these type of things. So these type of contributions that you have made to the society is really what drives us and what has led to a lot of these advances. So there are many, many ways that you can help, not just financially. We have about 1 minute left, and I just wanted to touch on one brief topic before I let you go. We talked about in this session many different types of new therapies, new ways of using therapies, and what I'd like you to walk out this door with is really a strong sense of hope, because this field is very strong right now, and there are lots of people interested in this, and there are lots of advancements that are going to be made. You can tell over the last 10 years how many new therapies have come to market, whereas in the first 50, 60 years of treating this disease, we had none and then just one. Remember this, we were using 3 pathways right now. There are over 150 pathways now that we know of that contribute to this disease, 150 pathways. Just imagine how many new therapies can be derived from looking at these individual pathways. So there's going to be a lot of information coming out. Stay tuned. Obviously, the PHA is the best way to keep your pulse, keep your fingers on the pulse of this. The PHA has wonderful sections on their websites talking about these new developments. So if you're computer savvy, you're set. If you're not, find a relative who's computer savvy, surf that site, and it really garners a lot of information from that. And with that, I'll let you guys go. I hope this was informative, and enjoy the rest of the meeting.